Welcome back to the deep dive. Today, we are stripping away the macroscopic world, the politics, the economy, the daily grind, and we are zooming in, way in.
To the microscopic level.
We are going to the front lines of a war that has been raging for, well, billions of years, a war that we humans only recently figured out how to join.
And you could argue it's the most important war we've ever fought. We're talking about the constant battle between the human host and the bacterial pathogen.
Specifically, we are talking about the weapons we use in that war, antibacterial agents. Now, I have the stack of research here, and we are anchoring this discussion heavily on Chapter 17 of Marie's Medical Microbiology.
That's our blueprint for today.
It is. That is our structural spine. We are following that map precisely. And then, for those high-yield clinical nuances, we're layering in wisdom from Chapter 28 of Jowett, Melnick, and Edelberg.
These are the absolute Bibles of the field. And let's be really clear about the mission today. The goal isn't to just, list a bunch of drug names.
If you want a list, you can go read a table in the textbook.
Right. Our goal is to understand the strategy, the tactics.
The logic of the kill. Because when you actually look at how these drugs work, it's not some kind of magic. It's engineering. It's tactical warfare.
It is. And it all hinges on one golden concept, something Paul Ehrlich proposed way back in the early 20th century. He had a great name for it. He called it selective toxicity.
Selective toxicity. I mean, it sounds simple, but when you really stop and think about it, it's a massive bioengineering challenge, isn't it?
Oh, it's incredible.
You need a substance that is absolutely lethal to a bacterium, but completely harmless, or at least, relatively harmless to the person swallowing it.
Exactly. And that is so incredibly difficult because at a cellular level, life looks a lot like life. We have DNA, bacteria have DNA, we have ribosomes, they have ribosomes.
We both have membranes.
We have membranes. So to achieve that selective toxicity, you have to find the tiny, almost invisible molecular differences, the chinks in their armor, and you have to strike right there with absolute precision.
Before we start launching missiles, though, I think we really need to appreciate the stakes here. We take antibiotics completely for granted now. You get a scratch, it gets a little red, you take a pill, you're fine.
It's just part of life.
But before this era,
it was a completely different universe. I mean, we call it the pre-antibiotic era, but that feels a little too clinical, a little too sterile.
It doesn't capture the fear.
It was a time of terror. A simple scratch from a rose thorn could lead to sepsis and death. A sore throat could progress to rheumatic heart disease. There's just no safety net.
You mentioned Ehrlich. He was famously looking for what he called the magic bullet.
He was. And he had some early success. Arsphenamine for syphilis, that was a start. But the real game changer, the moment the modern world truly began, was 1935. A German pathologist named Gerhard Domack.
This is the famous Prontosil story.
It is. It's a fantastic story. Domack was working with dyes for industrial purposes. He found this red dye, Prontosil, and he discovered something really bizarre.
What was that?
If you put it in a test tube with a bunch of bacteria, nothing happened. The bacteria just swam around happily.
Which is the opposite of what you'd expect.
Exactly. But if you gave that same dye to a mouse that had a lethal infection, the mouse lived.
That is a plot twist. Usually it works in the tube and then fails in the animal. Why did it work that way?
Because Prontosil was what we now call a prodrug. It wasn't the weapon itself. The host's own body, specifically the liver, had to metabolize the dye. It had to break it down.
And that breakdown product was the actual drug?
Yes. It broke down into sulfanilamide. That was the active agent. It was the very first sulfa drug.
And the legend has it he actually used this on his own daughter.
He did. It's an incredible story.
Okay.
She had a severe streptococcal infection in her arm, and she was facing either amputation or death. He injected her with this experimental red dye.
And she recovered.
She recovered. It's one of those, cinematic moments in science that really prove the concept of systemic chemotherapy.
And then just a few years later, the happy accident. The mold. Fleming.
The discovery of penicillin in 1928. Although it's important to remember it didn't become a viable medicine until Florian Chain scaled it up in the 1940s. But that discovery kicked off the golden age.
Suddenly we had everything.
All at once. Streptomycin, chloramphenicol, tetracyclines. It felt like we were invincible.
But, and this is really the theme of our entire discussion today, the enemy gets a vote.
The enemy always gets a vote. And you have to remember, bacteria reproduce maybe every 20 minutes. Their evolutionary clock is spinning 1,000,000 times faster than ours.
So as soon as we deployed penicillin,
they countered. Immediately, they learned how to destroy it. So what we're really discussing today isn't some static list of chemicals. It is a dynamic, ongoing arms race. It's a snapshot of an evolving chess match.
So let's get into the tactics. Let's act like military strategists for a bit. We need to identify the enemy's fortifications, their supply lines, and then figure out how to disrupt them.
Okay, sounds good. Let's start at the perimeter, the outer fortification, the cell wall.
This is section 1 of Murray, chapter 17, Inhibition of Cell Wall Synthesis. Now, why is the cell wall such a perfect target? I mean, humans have cells. Don't we have walls?
We do not. And that's the key to its selective toxicity. Human cells are, well, they're soft. We have a flexible cell membrane made of lipids, but that's it.
But bacteria are different.
Bacteria are under immense pressure. Literally, their internal osmotic pressure is sky high. Think of a bacterium like a bicycle tire that has been overinflated to the point where the rubber is screaming.
So the only thing keeping it from just popping is this rigid mesh cage on the outside.
Precisely, the peptidoglycan layer. It's their suit of armor.
Yeah.
So if you can compromise that mesh, if you just snip a few of those wires, the internal pressure does the work for you. The bacterium blows apart. It lysates.
And that makes cell wall inhibitors fundamentally bactericidal. They don't just stop growth. They actively kill the bug.
Correct. They kill.
So let's look at the mesh itself. Peptidoglycan. Murray describes it as this cross-linked polymer. What does that actually look like?
Imagine a chain link fence. That's a great mental model.
Yeah.
You have these long horizontal bars made of alternating sugars and acetylglucosamine and acetylmoramic acid.
Nag and NAM.
Megan and Nam. Sounds like a cartoon duo.
It does.
They just repeat nag, nam, over and over. But a stack of horizontal bars isn't a fence, right? It just falls over.
Yeah.
You need to tie them together vertically. You need cross links.
And that's where the peptido part of peptidoglycan comes in.
Right. Hanging off the NAM sugars are these little peptide chains. And the bacteria has specific enzymes. Think of them like construction workers that grab those peptide chains and link them together, building the fence.
These enzymes are called transpeptidases.
That's their technical name. But in the literature, they have a much more famous name.
Penicillin-binding proteins, PBPs.
We call them PBPs. Why? Because that's what penicillin binds to.
Okay, so we have this picture. The construction workers, the PBPs, are busily stitching the skunks together to keep the bacteria from exploding. Enter the first weapon, the beta-lactam.
The beta-lactam antibiotics, which is a huge family that includes penicillins, cephalosporins, carbapenems, and monobactams. They all share a specific chemical structure.
The beta-lactam ring.
That's the warhead. And this is the crucial bit of molecular espionage, the way it tricks the bacteria.
How does it do it?
It is the ultimate Trojan horse. That beta-lactam ring is a molecular mimic. It looks almost exactly like the dealanine sequence at the very end of the bacterial peptide chain.
Wait, explain that. So it looks just like the building material the worker is supposed to use.
Yes. The PBT enzyme is designed to grab that deala sequence to build the next link in the fence. The antibiotic floats by, and it's a perfect decoy. The PBP grabs the antibiotic instead.
She gets fooled.
Worse than fooled, it's a suicide trap. As soon as the PBP grabs the beta-lactam, the ring springs open and forms a covalent bond with the enzyme's active site.
Covalent meaning permanent.
Irreversible. The construction worker is now permanently handcuffed to a fake brick. He's useless. He's out of the game.
So construction of the wall starts. But does that alone kill the bacteria? Or does it just stop it from growing?
This is a massive exam bait point and a really critical clinical concept. Beta-lactams are bactericidal. They kill. But only if the bacteria are actively growing.
Walk me through that. Why only when they're growing?
Well, if a bacterium is just sitting there dormant, it isn't building a wall. It doesn't need its PBP, so you can throw all the penicillin you want at it. It won't care.
Okay.
But if it's growing, dividing, it needs to expand that wall. And to expand, it actually has to carefully cut the old mesh in order to insert new pieces. It uses enzymes called autolysins to make those cuts.
Ah, I see where this is going. So the autolysins are still cutting the mesh to make room for expansion?
But the PBPs are all handcuffed and can't add the new pieces to fill the gaps. So you have cutting without building. Structural integrity fails. Pop. The cell lyses.
That is elegant and brutal. But of course, the bacteria haven't taken this lying down. Murray outlines 3 main strategies they use to resist. Let's break down the enemy's countermoves.
Countermove number 1 is basically exclusion, the keep it out strategy. This is mostly a tactic for gram-negative bacteria.
So remind us of that fundamental structural difference between gram-positive and gram-negative.
Sure. Gram-positives have a very thick, exposed peptidoglycan wall on the outside. It's easy to get to. But gram-negatives have a thin wall, and they protect it with an extra layer, an outer membrane.
So for a drug to even reach the wall in a gram-negative, it has to get through that outer membrane first.
Right. It has to swim through these little protein channels called porins. So the bacteria's defense is simple. It just changes the locks.
How?
It mutates the genes for those porin proteins. Maybe it makes the hole smaller, or it changes the electrical charge of the channel.
Right.
Suddenly the antibiotic can't fit through anymore. It just bounces off the outer membrane.
Simple. Counter move number 2, disguise. Or as Murray calls it, altered target.
This is where the bacteria gets even smarter. The drug gets in, it reaches the construction site, but the construction worker, the PBP, looks different.
So the binding site itself is changed.
The binding site is altered. The drug tries to attach, but it doesn't fit anymore. It can't handcuff the enzyme.
This is the exact mechanism for one of the most famous superbugs, right?
It is. This is MRS, methicillin-resistant Staphylococcus aureus. Staph aureus acquired a brand new gene, it's called MECA, which codes for a completely new construction worker, a protein called PBP2A.
And our drugs can't bind to PBP2A.
Penicillin can't grab it. Methicillin can't grab it, so the bacteria just keeps building its wall, completely ignoring the antibiotic that's floating all around it.
And the third counter move, pure destruction, the missile defense system.
These are the beta-lactamases. These are enzymes that the bacteria manufactures and secretes. Their sole job is to hunt down the antibiotic molecule and slice that critical beta-lactam ring open.
And if you break the ring,
the drug is useless. It becomes totally inert. And this is where the arms race gets really, really heated. We develop penicillin. They develop penicillinase to destroy it.
So we developed methicillin to resist the penicillinase.
And they developed MRSA with an altered target. So we moved on to the cephalosporins.
And they developed ESBL's extended spectrum beta-lactamases that can kill those.
It is a constant back and forth.
Endless. Okay, let's look at our side of the arsenal in more detail. We have 4 main families of beta-lactams. Let's start with the originals, the penicillins.
Right, and we tend to group these by their spectrum, what kinds of bugs they can hit. You start with the natural penicillins, Pen G and Pen V. These are the OGs.
What are they good for?
They are still great for things like syphilis and basic strep infections, but they're fragile.
Fragile in what way?
Well, stomach acid destroys penicillin G, for example, so it has to be injected. And, more importantly, almost all Staph aureus you'll find in the world today makes penicillinase. So natural penicillin is basically useless against staph.
So in response to that, we built the penicillinase-resistant penicillins.
Exactly. Methicillin, oxalin, naphthalin. What the chemists did was they chemically added a big, bulky side chain to the molecule. It acts like a shield.
A shield for the ring.
Yes. The bacterial enzyme, the beta-lactamase, can't get close enough to cut the ring because this bulky group is in the way.
So these were designed as the anti-staph snipers.
They were. And for methicillin-susceptible staph, or MSSA, they're still the absolute drugs of choice. But of course, once MRSA appeared, they failed, because MRSA uses that altered target strategy, not the enzyme strategy.
Okay, then we wanted to hit more bugs. So we have the broad-spectrum penicillins, ampicillin, and amoxicillin.
For these, we tweak the structure to make them more hydrophilic, more water-loving. This helps them slip through those porin channels of gram-negative bacteria.
So suddenly we could hit things like E. coli.
Right. E. coli, Proteus, Salmonella. It opened up a whole new world of treatment.
But they are still vulnerable to the beta-lactamase enzymes, right?
Yeah.
Amoxicillin gets chewed up by those, just like regular penicillin does.
It does, which led to one of the absolute cleverest strategies in all of pharmacology, the bodyguard strategy.
I love this concept, the beta-lactamase inhibitors.
These are molecules like clavulanic acid, sulbactam, and tazobactam. They look a lot like penicillin, but they aren't really antibiotics on their own.
They're decoys.
They are sacrificial decoys. We mix them in with the real antibiotic.
So you take amoxicillin and you mix it with clavulanic acid, and that gives you the combination drug, Augmentin.
Exactly. So you swallow the pill. The bacteria sees the clavulanic acid, thinks it's a threat, and sends out its beta-lactamase enzymes to attack it.
But the inhibitor is a trap.
The enzyme binds to the clavulinate and gets stuck. It commits suicide on the decoy.
And in the meantime, the real antibiotic, the amoxicillin, slips past completely untouched to go and kill the bacteria.
It's a brilliant sacrificial play, a suicide inhibitor. And it works beautifully to restore and expand the spectrum of these older drugs.
All right, that's a great strategy. Let's move to family number 2, the cephalosporins.
These originally came from a mold called Cephalosporium. They are basically cousins to penicillin. They still have a beta-lactam ring, but they're inherently a bit tougher against those beta-lactamases, and we organize them by generations.
There's a very distinct pattern here that Murray highlights, and it's key for exams. As you go from the first generation up to the fourth generation, what generally happens to the coverage?
In general, you trade gram-positive coverage for gram-negative coverage, and you also tend to gain CNS penetration.
So a first gen is strong on gram positives, weak on gram negatives, and a third gen is strong on gram negatives, but maybe weaker on gram positives.
That's the classic rule of thumb. So first gen like cefazolin, which is given IV, or ceflexin, which you know as Kiplex, they're fantastic for skin infections. They nail staph and strep.
And what about gram negatives?
They had a few of the easy ones. We remember them by the mnemonic PEC, Proteus, E. coli, and Klebsiella.
Simple enough. Then second generation.
The second gen is kind of a confusing middle child. Drugs like cefoxitin or cefuroxime. They basically add a few more bugs to the list, like some anaerobes and Haemophilus influenza. So we sometimes call them the HEN PEC drugs.
And then we get to the third generation. These are the hospital workhorses.
Oh yeah, ceftriaxone, ceftazidime. These are the big guns for serious gram-negative infections. And a really key feature here is that they cross the blood-brain barrier much more effectively.
Which is something penicillins generally don't do very well.
Right, so if you have something like meningitis, an infection of the covering of the brain, ceftriaxone is a go-to drug. It's also the first-line treatment now for gonorrhea.
But there is a huge trap here for students regarding a specific nasty bug.
A massive trap, Pseudomonas aeruginosa. It's a highly resistant, very dangerous bug. Ceftriaxone does not kill it.
So what do you use?
If you suspect or have confirmed Pseudomonas, you need a different third gen, specifically ceftazidime, or you move up a generation.
To the fourth gen, cefepime.
Cefepime is sort of the have-your-cake-and-eat-it-too drug. It penetrates the gram-negative outer membrane incredibly well, so it hits Pseudomonas hard, but it also retains the excellent gram-positive activity of the first generation. It's very broad, very powerful antibiotic.
Now, Jabwetz throws in a very specific high-yield warning for some of the cephalosporins. Something about, of all things, alcohol.
Yes. A few cephalosporins, specifically those that have a chemical side chain called a methylthiotetrazole group, or MTT group, like cefotin, they block an enzyme in the human liver that breaks down alcohol.
So it causes a disulfiram reaction.
Exactly. If a patient on one of these drugs drinks a glass of wine, they'll accumulate a toxic metabolite called acetaldehyde. They'll get flushed, start vomiting, get a pounding headache. It's miserable.
Good to warn patients about.
And that same side chain can interfere with vitamin K metabolism and increase the risk of bleeding. It's rare, but it's a classic exam question.
Okay, family number 3, the carbapenems. Imipenem, meropenem.
If penicillin is a handgun and a cephalosporin is a rifle, the carbapenems are tactical nukes. They are the broadest spectrum beta-lactams we have by far.
What do they hit?
Pretty much everything. Gram-positives, gram-negatives, including Pseudomonas and anaerobes. They are our biggest guns.
So why on earth don't we just give them to everyone?
Stewardship. It's all about antibiotic stewardship. If you use the nuke for every minor skirmish, you're just breeding resistance to your last line of defense. We save these for the absolute sickest patients with confirmed multi-drug resistant infections.
And there's a specific quirk with imipenem, isn't there?
Yes, the kidney enzyme issue. The human kidney has an enzyme called dehydropeptidase that just loves to chew up and destroy imipenem. So if you just give the drug by itself, it never reaches the therapeutic level.
So we have to package it with something else.
We package it with a compound called cilastatin.
And cilastatin inhibits that kidney enzyme.
Yes. It has zero antibacterial effect. It's purely a bodyguard for the drug, protecting it from the human body. The mnemonic is imipenem lasts when given with cilastatin.
Clever. And our last beta-lactam family, the monobactams, Astrionem.
This one is the sniper. It has a very weird structure, just a single isolated ring. And because of that unique shape, it only binds to one specific PBP, PBP3, which is found only in gram-negative aerobes.
So it completely ignores gram-positives and anaerobes.
Completely ignores them. It's a very narrow spectrum.
That sounds limiting. Why would we use it?
One very important reason. Because the structure is so different from penicillin, it generally does not cross-react in patients with a severe penicillin allergy.
Ah, so it's a safety issue.
If a patient goes into anaphylactic shock with penicillin but has a serious gram-negative infection, Astrionem can be your life raft.
Okay, we've covered the beta-lactams in detail, but they aren't the only drugs that attack the wall. Let's move to Section 2. Non-beta-lactam cell wall inhibitors.
And the most famous one here, without a doubt, is vancomycin.
Vancomycin, the absolute go-to drug for MRSA.
It is. It's in a class called glycopeptides. And it is massive. Molecularly, it's a giant boulder compared to the little penicillin brick.
Right.
And because it's so huge, it cannot fit through those porous channels of gram-negative bacteria.
So it's physically impossible for vancomycin to kill something like E. coli.
Correct. It can't even get to the target. It is strictly for gram-positives.
So how does it work? It doesn't have a beta-lactam ring to trick the enzyme?
No, it uses a different strategy. Remember the ally-D-ala tail at the end of the peptide chain? Beta-lactams mimic it to fool the enzyme. Vancomycin uses brute force.
What does it do?
It just wraps itself around the D-ala tail itself.
It gives it a bear hug.
It gives it a tight bear hug. It's like putting a big, bulky cap on the end of a Lego brick.
Yeah.
Construction worker, the PBP, comes along to link that brick to the next one, but he can't get to the binding site because this giant vancomycin molecule is in the way. It sterically hinders the cross-linking.
So simple. So effective. But again, resistance. Viargius. Vancomycin-resistant Anaerocochi. How do you beat a bear hug?
You change the person being hugged. The bacteria cleverly mutate their genetic code to change the terminal D-alanine to a D-lactate.
D-ala becomes D-lac. That sounds like a tiny chemical change.
It is. It just removes a single potential hydrogen bond. But that is enough. Vancomycin tries to hug the new tail, but it can't get a good grip. It slips off. The binding affinity drops by a factor of 1,000.
But the PBP can still use it.
The PBP doesn't care.
Yeah.
It can still use the D-lactate tail to build the wall. The bacteria wins.
There is a classic side effect with vancomycin called red man syndrome. Tell us about that.
It's very dramatic. You start the IV infusion, and within minutes, the patient starts flushing bright red from the head and torso downwards. It looks exactly like a severe allergic reaction.
But is it a true allergy?
No. That's the key point. It's not an IgE-mediated anaphylactic reaction. It's a direct toxic effect of the drug on the body's mast cells. The drug makes them dump all their histamine at once.
And what causes that to happen?
Infusing the drug too fast.
So the fix isn't stop the drug forever.
The fix is slow down. You stop the infusion, maybe give an antihistamine like Benadryl, and then restart it at a much slower rate.
Good to know. What about a newer agent, daptomycin?
This is a really interesting one. It's a lipopeptide. It has this long, fatty lipid tail. It inserts that tail into the bacterial cell membrane like a dagger. Then it aggregates multiple daggers come together to form a pore or a channel.
So it pokes a hole directly in the membrane, in the balloon itself.
It causes a rapid depolarization of the membrane potential. All the ions leak out, the potential collapses, and DNA and RNA synthesis just grinds to a halt. The cell dies very quickly.
But there is a massive do not use warning for daptomycin in a specific clinical setting.
Absolutely. Do not use daptomycin for pneumonia.
Why not? It kills the bugs that cause pneumonia, right?
In a petri dish, yes. But in the human lungs, our alveoli are coated with surfactant, that slippery substance that keeps them from collapsing. For reasons we don't fully understand, surfactant binds to daptomycin and completely neutralizes it. It's like throwing a lit match into a bucket of water.
Fascinating. Let's briefly touch on the neosporin crew. Bacitracin and the polymyxins.
Bacitracin is another cell wall inhibitor. It blocks the lipid carrier, sort of the conveyor belt, that ferries the wall subunits across the membrane to the outside. It's way too toxic for your kidneys to take systemically, so we only use it in topical ointments.
And polymyxins like colistin.
These act like detergents. They're positively charged, and they bind to the negatively charged outer membrane of gram-negatives and just dissolve it, disrupting its integrity.
But they're also very toxic.
Historically, we stopped using them because they're terribly neurotoxic and nephrotoxic. They shred your kidneys.
But they are making a comeback.
Out of pure desperation, we now have multi-drug-resistant gram-negative bugs like Acinetobacter or Pseudomonas that are resistant to everything else. We are being forced to pull colistin off the shelf. We have to accept the high risk of kidney damage because the alternative is the patient dying from the infection.
Javats mentions a specific resistance gene here too, the MCR1 gene.
Yes, and this is terrifying. It's plasmid-mediated colistin resistance. That means it can be easily passed between different species of bacteria. If that gene spreads widely, we may truly have no drugs left for some of these infections.
And just to round out the wall, let's briefly mention the TB squad, the mycobacterial agents.
Right, just briefly, because TB is its own universe. But its cell wall is very unique. It's waxy, full of mycolic acid. So we use specific drugs, Isoniazid, or INH, inhibits the synthesis of those mycolic acids. Ethyl butyl inhibits a different component called arabinogalactan.
So they're all wall inhibitors, but for a very specific type of wall.
Exactly.
Okay, we have successfully breached the wall. We are inside the cytoplasm. Now let's go after the machinery. Section 3, the factory. Inhibition of protein synthesis.
We are targeting the ribosome.
Now hold on. I have ribosomes. You have ribosomes. Why doesn't a Z-Pak shut down my liver?
It's another beautiful example of selective toxicity. It's because your ribosomes are different. They're bigger. Bacterial ribosomes are classified as 70S ribosomes. Ours in our eukaryotic cells are 80S.
So it's a structural difference.
It's a structural difference. The drugs are like keys that only fit into the 70S lock, not the 80S one.
And that 70S ribosome has two main parts. The 30S subunit, which we can think of as the ground floor of the factory, and the 50S subunit, the upper deck. Let's start on the ground floor. 30S inhibitors.
The heavy hitters here are the aminoglycosides. Gentamicin, tobramycin, amikacin.
How do they work?
They bind irreversibly to the 30S subunit, and they mess with the decoding center. So when the mRNA instruction tape comes through the ribosome, it gets misread. The ribosome starts grabbing the wrong amino acids and stitching them together.
So the bacteria starts producing junk proteins.
Toxic, misfolded junk proteins that clog up the cell and actually damage the cell membrane. And this makes the aminoglycosides unique among this group.
How so?
Most protein synthesis inhibitors just stop growth. We call that bacteriostatic. Aminoglycosides, because they produce these toxic proteins, actually kill the bacteria. They are bactericidal.
But they have a very specific and very important entry requirement.
They do. They need an active transport pump to get across the inner cell membrane. And that pump is energy dependent. It's powered by an oxygen-dependent process.
So if you are an anaeroba, a bacteria that lives and thrives without oxygen,
You are naturally intrinsically immune. Aminoglycosides cannot touch you. The mnemonic is anaerobes don't breathe air, so aminoglycosides don't work there.
and the toxicity profile in humans?
Notorious. They tend to accumulate in two places, the inner ear and the kidney. So they cause ototoxicity and nephrotoxicity, and the hearing loss can be permanent.
Okay, the other major 30S drug is tetracycline, or more commonly now, doxycycline.
These are bacteriostatic. They don't cause misreading. They just block the docking port, the A site. The tRNA truck that's carrying the next amino acid tries to park, but doxycycline is sitting in its parking spot.
No parking, no protein.
Simple as that.
How do bacteria resist this?
Well, if you can't park your car because someone's in your spot, you tow the car. The bacteria develop efflux pumps. These are proteins in their membrane that recognize the drug and literally pump it back out of the cell as fast as it flows in.
And the classic side effect warning for kids and pregnant women?
Tetracyclines are chelators. They bind very strongly to calcium. If you give this to a young child whose teeth and bones are still developing, the drug will bind to the calcium in those tissues.
And what does that do?
It causes permanent brown discoloration of the teeth, and it can affect bone growth.
Let's move upstairs in the factory, the 50S inhibitors.
First up, the macrolides. This is your Z-Pak. Azithromycin, clarithromycin, erythromycin.
What's their mechanism?
They bind to the 50S subunit, and they block the exit tunnel. So the protein chain starts to get built, but as it gets longer, it runs into the drug and gets stuck. It's like plugging the exhaust pipe of a car. The engine stalls.
And how do bacteria fight back?
The most common way is methylation. The bacteria acquire a gene, usually called an gene, for an enzyme that sticks a little methyl group onto the ribosome at the drug's binding site. It just changes the shape of the tunnel, and the drug can't bind anymore.
Okay, next up, clindamycin.
Clindamycin works in a very similar way to macrolides, blocking that exit tunnel. It's fantastic for anaerobes, the very ones that gentamicin couldn't touch. So we use it a lot for things like aspiration pneumonia or abdominal infections.
But it has a bit of a dark reputation.
It does. Clindamycin is very, very good at killing off the good bacteria that live in your gut. And when you wipe out all the competition, a dormant bad guy can sometimes wake up and take over.
Clostridioides difficile, C. diff.
C. diff. It can overgrow and cause a severe, sometimes life-threatening colitis.
What about a newer drug, linezolid?
Linezolid is in a unique class called the oxazolidinones. It works differently from all the others. It actually stops the two floors of the factory, the 30S and 50S subunits, from even coming together in the first place.
It blocks the initiation complex.
Exactly. And because its mechanism is so unique, it works on many of the bugs that are resistant to everything else, like VRE and MRSA. It's a true heavy weapon.
And finally, a drug from the history books, chloramphenicol.
We rarely use it now, but it's important historically. It blocks the actual stitching reaction. the enzyme called peptidyltransferase. It's powerful, but it's very toxic to human bone marrow.
It causes aplastic anemia.
It can. And in premature babies who lack a specific liver enzyme called glucuronidase, they can't metabolize the drug.
What happens to them?
The drug builds up to toxic levels. They turn ashen, limp, and they die of cardiovascular collapse. It's called gray baby syndrome.
So we avoid that one unless we have absolutely no other choice. Okay, Section 4. We're going deeper. Into the command center, the nucleoid. We're talking inhibition of nucleic acid synthesis.
We are attacking the blueprints, the DNA itself. The stars of this show are the fluoroquinolones, ciprofloxacin, levofloxacin.
Then these famously target DNA gyrase. What on earth is a gyrase?
Okay, imagine you have a long, twisted rope. That's the DNA double helix. If you try to pull the two strands apart in the middle to copy them, the rope ahead of you gets tighter and tighter and tighter. It knots up. That's called supercoiling.
So gyrase is the untangler.
Yes. This is a topoisomerase.
Yeah.
Its job is to make a strategic cut in the DNA, pass one strand through the other to relieve the tension, and then reseal the cut. It's essential for replication.
And the quinolone drug?
It's a poison. It binds to the gyrase enzyme while it's holding the cut ends of the DNA. It freezes the enzyme in the middle of its SNP.
So the DNA is left permanently cut?
Yes. The chromosome fragments and snaps. It's absolutely catastrophic for the cell.
But these drugs have a really weird connective tissue side effect in humans.
They do. We don't fully understand the mechanism, but they're associated with causing tendinitis and even spontaneous tendon rupture. The Achilles tendon is the classic one.
Cipro snaps tendons.
It's a good mnemonic.
Yeah.
And because of concerns about cartilage damage in animal studies, we generally avoid them in pregnant women and young children.
What about rifampin?
Rifampin targets a different enzyme. It goes after RNA polymerase. It stops the transcription of DNA into messenger RNA. No transcription, no proteins, no life.
And its main uses?
It's a cornerstone of tuberculosis treatment, and we also use it for prophylaxis against meningococcal meningitis.
And the famous clinical pearl?
It turns all your body fluids a harmless but dramatic bright orange-red color. Urine, sweat, and even tears. You have to warn patients, or they will be absolutely terrified.
Okay, what about metronidazole?
This is the designated assassin for anaerobes and certain protozoa. It's another prodrug. It is completely harmless until it enters an anaerobic environment.
What happens inside the anaerobe?
These organisms have specific enzymes, like nitroreductases, that are part of their metabolism. These enzymes reduce the drug. They donate an electron to it.
And that activates it.
It turns it into a highly reactive, toxic free radical. This radical then just acts like a shredder. It attacks the bacterial DNA and breaks it into pieces.
And since we are aerobic and don't have those enzymes,
we are safe. It's an incredibly selective mechanism. We use it for the get-gap infections, Giardia, Entamoeba, Trichomonas, Gardnerella, anaerobes, and H. pylori.
Excellent. Finally, Section 5, the supply line, the anti-metabolites.
This is all about starving the army, specifically starving them of folic acid.
Why is that a good target?
Because bacteria need folic acid to make the building blocks of DNA, the nucleotides. But here's the trick. Humans eat folate. We get it from salad and fortified grains. Bacteria are too proud, or their membranes are too impermeable. They can't absorb it from the environment. They have to make it from scratch.
So we sabotage their unique factory line.
Exactly. The raw material they start with is a molecule called PEBA. The first drug, a sulfonamide, looks almost identical to PEBA. It's a competitive inhibitor. It jams the first enzyme in the chain, dihydroapatite synthase.
And the second drug in the combo, trimethoprim.
It jams the next enzyme in the production line, an enzyme called dihydrofolate reductase.
And that's why we almost always use them together, as Bactrim or TMP-SMX.
It's a synergistic combination, a sequential blockade. If you just block one step, the bacteria might be able to ramp up production and push through. But if you block two steps, one after the other, the whole line grinds to a halt. It's also much harder for bacteria to simultaneously mutate two different enzymes to become resistant.
We have covered a staggering amount of ground, from the cell wall to the ribosome to the DNA itself.
It's a complete tactical map of antibacterial warfare.
So let's see if we can use it. It's time for Section 6, the quiz.
I'm ready. Put me in the hot seat. Or even better, let's put the listener in the hot seat.
Here's the deal. I'm going to present a short clinical scenario. You, our listener, try to come up with the answer in your head. Then our expert will break down the reasoning. Ready?
Let's do it.
Scenario 1. You have a patient with a classic case of walking pneumonia, and the lab confirms it's caused by mycoplasma pneumonia. You smartly prescribe penicillin. The patient comes back a week later, even worse. Why did the drug fail?
Pause out there. The answer is target absent.
Explain that.
Beta-lactams like penicillin work by attacking the peptidoglycan cell wall. Mycoplasma is a very weird genus of bacteria that does not have a cell wall. It only has a cholesterol-containing membrane. You can't bomb a bridge that doesn't exist. The drug had nothing to target.
A perfect example of knowing your bug. Okay, scenario 2. You are treating a patient with a very serious MRSA bloodstream infection. You start an IV infusion. 10 minutes later, the nurse calls you in a panic. The patient is bright red from the neck up, itching, and feels hot. Which drug is it, and did you just cause a life-threatening allergic reaction?
Pause that the drug is almost certainly vancomycin. And this is the classic picture of red man syndrome.
And is it an allergy? Did you kill them?
No and no. It is not a true IG-mediated allergy. It's a direct histamine release from mast cells caused by infusing the drug too quickly. You should stop the drip, give an antihistamine, and then restart it at a much, much slower rate.
Scenario 3. We need to kill a gram-negative bug that's causing sepsis. For this question, we want to use a drug that inhibits protein synthesis and is bactericidal. But the source of the infection is a large, deep abscess with essentially zero oxygen inside it. Which drug fits the bill?
Pause. That's a trick question. Based on those criteria, there isn't one.
Why not?
The only class of protein synthesis inhibitors that are reliably bactericidal are the aminoglycosides. But what's their rule? They require oxygen for their active transport pump to get into the cell. In an anaerobic abscess, they're completely useless. You would need to surgically drain the abscess or choose a different class of antibiotic entirely.
Excellent point. Scenario 4. A 25-year-old college athlete is being treated for a complicated UTI. During soccer practice, she plants her foot to make a cut and feels a sudden, sharp pop in her heel. Her Achilles tendon has ruptured. What class of antibiotic was she most likely taking?
Pause. That has to be a fluoroquinolone, like ciprofloxacin or levofloxacin.
It's a rare side effect, but it's a classic board question. Tendinopathy and tendon rupture. All right, last one. Scenario 5. A patient is taking a Z-Pak, which is azithromycin, for a respiratory infection. Which floor of the ribosome factory is being shut down? The 30S or the 50S subunit?
Pause azithromycin is a macrolide, so that's the 50S subunit.
Yeah.
Macrolides, clindamycin, they all bind to the big subunit and block that protein exit tunnel.
If you got those right at home, you're thinking like a microbiologist. If not, go back and really visualize the mechanism. And don't just memorize drug A does B. Visualize the molecular machine and what's breaking.
That is the absolute key. If you can see the machine in your mind's eye, then the spectrum of activity, the side effects, and the resistance mechanisms all just make logical sense.
Before we sign off, I want to leave you with a thought. We called this whole discussion an arms race.
It is. And to be perfectly honest, right now, we're losing.
How so?
We haven't discovered a truly new class of antibiotics, a fundamentally new mechanism of action, in decades, not since the 80s. Meanwhile, bacteria are out there sharing resistance genes on plasmids like they're trading cards.
So all this talk about stewardship isn't just hospital bureaucracy.
It's species survival. If we burn through these precious drugs, prescribing them for viral colds, using them indiscriminately in livestock feed, we are effectively disarming ourselves for the future.
Imagine going back to the year 1930, a world where a routine C-section or a hip replacement is a massive gamble with death because of the risk of a post-operative infection. That's the post-antibiotic era we are trying to avoid.
So treat these molecules with respect. They are precious, and they are arguably non-renewable resources.
And one more thing for you to chew on. We just spent an hour talking about all the clever ways we've devised to kill bacteria. But your body is mostly bacteria? The microbiome in your gut, on your skin?
Trillions of them.
So when you drop a nuclear bomb like a carbapenem, or even a broad-spectrum penicillin into that system, what happens to the complex society of friendly bacteria that we now know regulate your immune system, your mood, your digestion? We are only just beginning to understand the immense collateral damage of this war.
It's a fascinating new frontier. Selective toxicity saves us from the acute death of an infection, but we're still learning about the long-term cost of that chronic disruption.
Something to think about. That's it for this deep dive. To consolidate all this information, we strongly encourage you to review Murray Chapter 17 and pay special attention to the tables on mechanisms of action and the boxes on resistance. Thanks for listening.
Stay curious.